Today's lecture is going to cover three things. We have the integration of metabolism, tying things together for you. We'll look at some recurring patterns in here where different pathways intersect, mainly at three crossroads of glucose-6-phosphate, pyruvate, and acetyl-CoA. And then we'll have the effects of how metabolism adjusts to exercise. We've already talked about how excess ethanol kind of looks like in the cell, looks like a diabetic cell. And then we'll talk about the glyoxylate cycle, which is something unusual we haven't done yet. And one of the rules is going to get broken through that cycle. So the first thing we want to do is take all those pathways that you've learned individually and try to tie them together. So I found this nice drawing that kind of shows that. It might be difficult to read the text, but at the bottom it says the heavy arrows indicate irreversible reactions. So everywhere you see a bunch of arrows is the reactions we've done. We've covered every one of these. And the heavy bolded arrows are the ones that are irreversible. So looking at here, you see the three in glycolysis that are irreversible, the reactions of gluconeogenesis going back that are irreversible. We can convert acetyl-CoA into fats on the right, which is very easy to do. We can also convert fats back into acetyl-CoA. That's totally a reversible process by different enzymes, but you can convert back and forth. We cannot convert acetyl-CoA back into pyruvate, not a net conversion. And we cannot convert pyruvate back into glucose net, right? We said those were irreversible steps. So once you go from glucose to glucose-6-phosphate, it's very difficult to get back to glucose unless you're a liver cell, right, to cut off that phosphate. And once you go through glycolysis to pyruvate, you could go lots of places from pyruvate. You can turn it into several amino acids. You can go back to glucose if you want. But once you step over to acetyl-CoA, there's no going back, right? Once you turn pyruvate into acetyl-CoA, you can't go back onto the sugar side of the tree. You can enter a citric acid cycle. You can turn into fats and so forth and make some amino acids. But you can't go back to the sugar side of the tree. And so let's talk about some of these recurring things or patterns that you see in this whole system. Okay, so the first thing we want to talk about is the difference between anabolism and catabolism. Anabolism was the process of building something, right? Assembling a molecule. And we generally do reduction type reactions or assembly type reactions, right? So either we make a new carbon-carbon bond or we reduce a double bond or something like that, right? In the other case, in catabolism, we generally are oxidizing things or breaking carbon-carbon bonds, right? So to do those two pathways, you need some kind of cofactor or other substrate, effectively. So in anabolic pathways, we said we generally use, in eukaryotes, NADPH and not NADH. NADH in prokaryotes and so forth. But in catabolic pathways, we tend to use NAD+, right? There's no need for NADP+ there because we want to keep them separated. We want to use the phosphorylated one, the NADP, in the case of anabolic systems, anabolic pathways, and the NAD+, the non-phosphorylated one, in the catabolic pathways. Occasionally we'll use FAD, yes, and we talked about when that would be appropriate, but we don't use the NADP for most catabolic systems. Okay, so a nice distinction is the phosphorylated version in anabolic reactions and the non-phosphorylated, the NAD, in catabolics. Now there are some exceptions. Right, you remember we had the first one we talked about in gluconeogenesis. Our only redox reaction in that whole pathway, step 6 of glycolysis, totally reversible. And since the enzyme already existed and it was reversible, there was no need to invent a new enzyme and use NADPH there. So we just use NADH. And that's probably because we inherited that ancient pathway from a prokaryote, which did not have NADP. Yeah, but in most of our newer pathways, or pathways unique to eukaryotes, we'll use NADP for anabolism and NAD+ for catabolism. I know we equated the two in terms of ATP. Each one's worth about 2.5 ATPs worth of energy, but they are not the same molecules. We have separate pools of these two. It's difficult to convert one to the other. It's a phosphorylation event, which would, of course, cost an ATP. But the pools of NADPH and pools of NADH are kept separate, right? So the ratio of one to the other can give the cell an idea of what state it's in. Is it in an anabolic state or is it in a catabolic state? On the right, we have three other things we can do to regulate enzymes. We can put them in different compartments, right? So we can put one in the matrix, we can put one in the cytoplasm, and those two pathways could be completely opposite of each other, such as we put glycolysis in the cytoplasm, but we tied gluconeogenesis to having to enter the matrix. So they weren't happening simultaneously in the same compartment. Fatty acid synthesis, totally in the cytoplasm, right? We just went over that last time. And then we do fatty acid oxidation, turning it into acetyl-CoA, completely in the matrix. So the two compartments separate the pathways, right? That's not possible in bacteria as much, but in eukaryotes, we can compartmentalize things. Even so, in higher organisms, we can compartmentalize things by organs or cells themselves, right? The liver does a lot of things the muscle cells don't do. Muscle cells do things that nerve cells don't do, and so forth. So we can specialize our organs when you get to multicellular or multi-system organisms. The other thing is enzyme concentrations. If you want to do two opposing pathways in the same compartment, you can upregulate the concentration of one enzyme and downregulate the opposing enzyme, and that would favor the one with the higher concentration. So you can do it by several ways. And of course, if you're a bacterium, that's kind of your only way to do it, is concentrations are inhibiting enzymes. How do you inhibit those enzymes? That's what's on the bottom left. You can inhibit them with allosteric interactions, so something is binding generally non-covalently in another pocket, usually other than the active site. We did this a lot with our ribonucleotide reductase. It was highly allosterically regulated. Remember all the triphosphorylated versions that bound in that activity site and told the active site what to do? We also had a lot of modifications that were covalent, such as phosphorylation, like shown underneath. We put an ATP, donates a phosphate to a serine. Also on the right, we had something, adenylation. Still used an ATP, but we attached the other part of the molecule. The AMP is actually attached, in this case shown on a tyrosine. So we did this with not only ATP, but UTPs and GTP and so forth. So we can modify it by a non-covalent interaction, or we can modify it by a covalent. These are generally post-translational modifications. Right, so an example of this is at glycolysis, right? So when we did the third step of glycolysis, converting fructose 6-phosphate to the diphosphorylated version of fructose, we used the enzyme PFK1. And we learned all about this in glycolysis and in how we had to bypass it for gluconeogenesis. But what I'm focusing on here is the regulation of it. So you learned this before. It's activated by an absence of ATP, that's AMP, right? And it's inhibited if ATP is abundant, which makes sense because generally you have molecules that inhibit their own production. So glycolysis is in the pathway or multiple pathways that lead to production of ATP. And along the way, we made a citrate at the beginning of the citric acid cycle. Both of those are indicating that we have an abundance of our products. So let's slow down this glycolysis mechanism. If ATP is not abundant, how do we detect it? Well, we detect its absence by the alternative molecule, AMP. ADP, you remember, two of those could be converted to an ATP and an AMP by your favorite little enzyme. And then you have AMP in abundance. It indicates a lack of ATP. So that would regulate this enzyme to work faster. So it's just an example of that, how you can use modifications by other molecules, not necessarily covalent modifications, just binding by other molecules to make something work faster or slower. Okay. Other examples, I don't have pictures for these, but I'll explain them to you. On the top left, we had our citric acid cycle. So the purpose of the citric acid cycle was to do oxidation events to generate a lot of electrons, and those electrons they don't exist in isolation. They're going to be transferred down the electron transport chain and ultimately to oxygen, so we can generate a gradient of protons to make ATP. If we're doing citric acid cycle and also not making ATP, one is generating electrons that the other is not using. So there's a backup of electrons there. That will shut the citric acid cycle down. If you're making ATP at the same time that you're using electron extraction in the citric acid cycle, then they complement one another. So one has to occur with the other. They occur synchronously. So if you're making ATP, you must be extracting electrons from the citric acid cycle. They occur together. So that makes sense that an abundance of ATP, just like with glycolysis is PFK, would inhibit the citric acid cycle. Because if I don't need to make it, let's not extract the electrons as rigorously. So ATP inhibits the reversible enzymes of our citric acid cycle. That's steps 3 and 4, our isocitrate dehydrogenase and the alpha-ketoglutarate dehydrogenase. Okay. I leave the last line underneath when I get to gluconeogenesis, but looking on the right, in our pentose phosphate pathway, remember we had 2 phases of this. One was the oxidative phase, where we converted a 6-carbon molecule into a 5-carbon molecule, and we lost the CO2, right? So that's kind of irreversible. And along the way, I got an NADPH out of it. Maybe that's the goal. I needed a lot of NADPH, or I needed a lot of 5-carbon sugars with phosphates on them for DNA synthesis or RNA synthesis. Alternatively, I could do the non-oxidative phase, which doesn't have a loss of carbons, right? It's a net conversion of those 30 carbons, right? We had 5 6-carbon sugars converted into 6 5-carbon sugars, or vice versa. So I don't lose any carbons. So doing the non-oxidative phase must be a reason that I don't want to lose or waste carbons. Whereas in the other one, I have an abundance of carbon. I don't mind wasting it to get to my 5-carbon sugar if I need NADPH reducing equivalents. So we had multiple modes we could use there. And so they're regulated, which mode I use, by what I need in the cell. Underneath, we have gluconeogenesis, right? So it's kind of the opposite process of glycolysis. It doesn't use the same enzymes, obviously, because we had some irreversible steps. But when I'm doing one, I want to downregulate the other, right? So this is controlled by hormones and by phosphorylations, as well as allosteric events with ATP and AMP and so forth. The citric acid cycle has a play on gluconeogenesis and glycolysis, because the first step of gluconeogenesis was to convert pyruvate into oxaloacetate. That was a carboxylation that happened in the matrix. Well, that was one of our steps of citric acid cycle, just run in reverse at that. I take the oxaloacetate and then reduce it to malate, so I can export it to continue gluconeogenesis. Right? If I didn't have to do that, there would be no connection between the citric acid cycle and gluconeogenesis. So because of that, I have control over it. Right? So the molecules that control the citric acid cycle inherently also control gluconeogenesis. Here's an example of those compartmentalization things. On the left, we have acetyl-CoA being converted to malonyl-CoA. And if you recall, this was the first step in fatty acid synthesis, where we used the acetyl-CoA carboxylase. We had the biotin carboxylase component and the carboxyltransferase component. And that enzyme is active when we want to make fatty acids. Well, of course, to make fatty acids, I must have an abundance of acetyl-CoA. If acetyl-CoA is abundant, I must be well fed, right? The cell's not lacking in something. Either it's got an abundant source of glucose, it's got an abundant source of another sugar, or something like that. If the sugars are not abundant, I don't want to convert my acetyl-CoA into fats. I want to convert fats into acetyl-CoA for food, right? So I don't want to be storing them. So this enzyme gets shut down if there's lots of or a lack of sugars and there's lots of fats around. On the other side, if you're, instead of making fatty acids, you're degrading them, which happens in the matrix, I want that to stop if I'm producing malonyl-CoA. Because if I'm making fats, I don't want to turn around and degrade them, right? Because it costs more to make them than you get out of it, just like with sugars. Right? So these two are compartmentalized and also inhibited by the other one's product. Right? Looking backwards at that large picture again, I had 3 junctions or 3 key molecules I pointed out. It was glucose 6-phosphate, pyruvate, and acetyl-CoA. And they're mainly surrounded by some irreversible steps. So looking at the picture on the left, converting glucose to glucose 6-phosphate, yes, that's an irreversible step in each direction by different enzymes, but we let that one go. Remember, we didn't really control that one so much just because it keeps the glucose in the cell. So we're going to forgive the cell for using that one. The only way you convert glucose 6-phosphate back to glucose is if you want to export it, and that's mainly done by the liver. The other 3 fates there of our glucose 6-phosphate are right down the center, we're going to do glycolysis. To the left, we're going to store it as glycogen. And to the right, maybe I don't need to store it. I have enough glycogen, and I don't need it for fuel right now. I'm doing well. I have enough ATP. So let's divide. The cell's healthy. It's got a lot of glycogen stores. ATP is abundant. Let's divide. Right? So the cell can produce more RNA, so it turns it into the ribulose 5-phosphate through the oxidative phase of the pentose phosphate pathway. Yes, I know it should say ribulose 5-phosphate, but you can easily convert that to ribose 5-phosphate. So they just showed that in the figure. On the right, farther down the pathway, we have pyruvate, and here's another junction where we can convert it to a couple amino acids. We can convert it to oxaloacetate if you want to make more glucose, or we can convert it to acetyl-CoA if I want to use it as fuel, as for energy. So two of those steps, converting it to acetyl-CoA and to oxaloacetate, are irreversible. So you have to choose, should you do that or not, right? And again, we just went over all the steps along the way to tell it whether or not to do this pathway. Once you convert it to acetyl-CoA, again, there's no going back. We can oxidize that in the citric acid cycle right down the center to CO2. We can store it as fatty acids on the right, or we can store it as ketone bodies on the left. Right? So converting it to ketone bodies, however, is irreversible. Converting it to CO2, irreversible. But the storage form, totally reversible. So if you look back at these three key junctures, the glucose on the left, the sugar side of the pathway, can be stored as glycogen. The pyruvate can effectively be stored as amino acids, some of the amino acids, and the acetyl-CoA can be stored as fatty acids. So we have storage forms of each of these, and then we have irreversible pathways from each of them. Right, the key here is you can't go backwards. We can't convert acetyl-CoA back to pyruvate. Okay? So how does this affect organisms such as humans, right? So on the graph on the top left, this is what happens in the body, not necessarily any individual cell or any individual organ, but overall, mostly muscles we're talking about here, but overall, how does the body respond to exercise or using muscles? So if you look at the graph, tick marks at the bottom, if you exercise only for a few hours, right, to even a couple of days, and then you just stop, what happens is you do these exercises is the mRNA spikes, right? In the muscles, the mRNA is produced rapidly. And then that's to tell the cell to make certain proteins. And then the exercise or the stress is over, and the mRNA gets degraded. Those extra proteins don't seem necessary anymore, and they get degraded. So overall, the protein content doesn't stay high. It goes back to a normal, steady state amount. But if you keep exercising and do that over weeks to months, the amount of mRNA in the cell tends to rise, right? It stays at a higher not much higher, but a higher steady state concentration. Well, with a higher amount of mRNA present all the time, you get more protein present. And of course, with more proteins, most of those are enzymes at this point, they can function better. So there's more protein, such as actin and myosin. There's more enzymes as well to do all these metabolic pathways like citric acid cycle. And you're going to produce more mitochondria as well. So the green at the top tells you the performance of that muscle or that organ. It'll be improved if you continually stress it over hours, days, months, and weeks into months. It adapts to this repeated stress or exercise. And so that's why it's hard to see an improvement if you only exercise for a couple of days, and a lot of people will quit going to the gym at that point because they don't see improvement. But if you do it for weeks to months, you'll definitely notice an improvement. But you have to stress the cells. On the right, I modified this from the one you have, I think. I added another spot to it. But on the right, we have this, I don't know how old it is, but these times have probably been improved since I grabbed this graph all those years ago. But the 100-meter sprint, just under 10 seconds, would probably be getting better than that. The 1 kilometer, if you want to call it a sprint, all right, just over 2 minutes. Right, you're going to run a marathon in a little over 2 hours. And the one on the right was done a little differently. It's how far can you run in 24 hours. So it's still a distance and a time. If you look at the average speed for these events, you get a little over 10 meters per second. The first one, a little under, 7 to 8 or so meters per second. The second one, 5.5 for a marathon, and then down to 3.5 for that very long endurance race. So over time, you can't run as fast for as long. It kind of makes sense. So what's the point of that? What do we get out of this graph? Well, it has a general downward trend. It's not quite linear. You notice there's some bumps in the graph. But if we take each part of it and look at why the points fall where they are, we can get some idea of what fuel the cell's using. In the very beginning, whether you run the 100 meters or the 200 meters, the times don't alter very much. right? The speed is still pretty much the same. So we're not exhausting all of our fuel, all of our immediate fuel in that 100 meters. So getting close to 200 meters, we might be running out of that immediate fuel. And after that, you see it drops when we get to, say, 1,000 meters, or even 300 meters or something. So what's going on? We must have an immediate reservoir of fuel that can only last so long. We talked about this the first day of class. Right? Our muscles have a supply of ATP, right? Roughly 223 millimoles of phosphate available. But we also have that creatine phosphate, which has about twice that much phosphate available. So we run out of ATP rather rapidly within seconds. The creatine phosphate can phosphorylate the ADP, and I get another few seconds of running out of that. So maybe up to 10, 20 seconds of fuel available, right, for those very fast sprints. And that's why you can run at 10 meters per second there. Well, not me, but Usain Bolt can. Right? But once we get past that, you notice the time gets slower, time gets longer. So if I put a trend line to that first part, right, it fits this line fairly well. So there's something going on here. This is likely the conversion of glycogen into fuel. Glycogen being mobilized into glucose. We converted glucose then into lactate, send that as fermentation back to the liver, and we convert the glycogen to CO2 as well. So this is glycogen stores mainly by the muscle itself, and it can keep this up for several minutes, maybe closer to, I don't know, half an hour or something like that. But you notice at some point the curve flattens, right? And we get a slower speed down here, and that's the blue trend line. So we get a slower speed. This is more likely conversion of glycogen from not the muscle, but delivered via the bloodstream from the liver, right? Same thing can be said about adipose tissue breaking down and then mobilizing that as ketone bodies, right? And also using that by the muscles. So the fats, there's a large abundance of phosphate available, right? Not in the phosphate, but potential phosphates available if we break down fatty acids. Same thing with glycogen, except we have to transport it from the liver to the muscle. So if you look at their maximum rates there of ATP production, those numbers kind of mimic the slopes I'm generating, right? So in the beginning, it's very large, 73.3 millimoles per second of ATP. So I can maintain that speed. But it slows down after that when I revert to using glycogen stores. I have to mobilize those stores. So it's a slower rate. That's the red line. And then it goes really slow, around 6 millimoles per second when you're in just longer distance running. And so at some point, I figured there has to be a limit. How far can you run? And I tried to find a point that wasn't on this graph originally, and that was the 24-hour continuous run. They could run 303 kilometers, which is quite amazing. Right? But it's still slow, right? It's only 3.5 meters per second. But it's not incredibly slow compared to the last block. So I think it's still running off of fatty acid stores. At some point, I think the limit to how far we could run would be physical damage to tissues and organs. Right? You could probably keep that 3.5 meters per second up for quite a long time. It's a little faster than walking. Right? So you probably can keep that up until you suffer some fatigue from organ or tissue damage. Right, and the last thing I want to talk about, I'm not going to mention the ethanol part too much because we already did that one, but I want to talk about how you bypass the citric acid cycle or find a way across the citric acid cycle. This is called the glyoxylate cycle, right, or how to break the rules. So plants do this in their seeds, and some microbes do this. Some bacteria can do this. So mainly, we're going to talk about how it happens in plants and then in one particular bacterium. So in plants, they do this in their seeds underground when they have no access to photosynthesis. Right? The seed is germinating below the surface in the soil, no light whatsoever. So how does it get energy? Well, we know the plants don't eat anything. The seeds don't eat anything for sure. They have a store of fats. They might have a few sugars around, but not very many. Sugars don't store long term. Fats can. So if you've ever eaten a lot of like peanuts or seeds or sunflowers or things like that, castor beans, they're loaded in fats, right, in oils. Right? So how do they convert those oils into useful fuel? Well, you can turn them all into acetyl-CoA, you know that, but you can't make sugars from acetyl-CoA. And I can't make new RNA, DNA without a sugar, without making glucose. Right, or at least fructose. So I need to be able to convert a fat, acetyl-CoA, back into a sugar, pyruvate. Well, we said we couldn't do that. It was irreversible. So we need a way around that. And that's where this glyoxylate cycle comes in. So we're going to hijack the cycle at the point of isocitrate. So you see isocitrate takes a detour through the middle of the circle here and becomes succinate and glyoxylate, right? And it's just a lyase. All we're doing is breaking a bond, right? No redox going on here. And that's shown in the little inset on the bottom right. We have some general base deprotonate the only OH on the isocitrate, right? That collapses to form a carbonyl, breaking the bond between carbons 2 and 3. Right? They labeled them 1, 2, 3 on the left, so on their label it's between 1 and 2, but it's technically carbons 2 and 3. Right? And then when that bond breaks, it grabs a proton from a neighboring donating base, general base up there called acid called AH. And then that leaves you a succinate and a glyoxylate. Right? So glyoxylate is 2 carbons long, succinate is 4 carbons long, and that accounts for all our carbons from isocitrate. So we didn't lose anything. What can happen now is the glyoxylate can be converted to malate. If you see in the figure on the left, we use another acetyl-CoA for that. So this is a transferase. We're going to take a 2-carbon group and put it on glyoxylate to make malate, right? Just like we do with citrate synthase. It's almost the same reaction. Okay? It's just putting it on a smaller piece. So 2 plus 2 makes 4 here, instead of 2 plus 4 making 6. But then now I have malate. I can reduce it once, and I have oxaloacetate. And I can continue through the citric acid cycle and end up with another isocitrate. What was the point of this? Is that useful or not? Well, I do get a succinate molecule out of this. Right? So on the next slide, I'm showing a modified version of that where I'm only leading you through the center pathway. So starting at the top again, just like a normal citric acid cycle, I have my oxaloacetate and acetyl-CoA. Citrate synthase assembles the citrate. We turn it into isocitrate. They inverted the image for you here. Number 1 would be on the far left of isocitrate in this case, instead of the far right. But we do the same reaction, the isocitrate lyase, and it becomes glyoxylate plus I liberate a succinate. The glyoxylate gets an acetyl group attached by the malic enzyme, or malate synthase. It becomes malate. I reduce it oxidize it to oxaloacetate, and I can start again. So effectively, if I continue this cycle, each round I convert 2 acetyl-CoAs into a succinate. Right? We couldn't do that before. We couldn't turn acetyl-CoAs into a 4-carbon molecule, right? Not one that we can use to make sugars. But succinate can easily be converted to a sugar, right? We just finish the citric acid cycle, turn it into oxaloacetate normally, and then we can turn that into PEP, right? We exit it out of the mitochondria through malate, oxidize it back to oxaloacetate, and then turn it into PEP, and from there you can make pyruvate or glucose. So yes, we are getting a net production of a pyruvate, or ultimately glucose, by using this pathway. So why don't we do this? Right? So this requires the enzyme isocitrate lyase, right? The one that we first talked about here, turning isocitrate into its 2 products. So what rule is this breaking? Well, for animals, you can't convert fats back into sugars. But seeds must do this. They can't make sugars any other way. They don't yet have access to photosynthesis until they finally sprout and get a leaf so they can make sugars that way. So if they're deep underground and they exhaust their supply of lipids using this method to make sugars so they can grow and divide and make RNA and so forth, if they do that so deep underground that they never get above the surface and make that first leaf before they run out of fats, they're not going to survive. And if they're close enough to the surface, then they will be able to. If they're too close to the surface, they're likely to get eaten by a rodent or something else. So the depth of the seed matters a lot for a plant to survive. Okay, so let's talk about that enzyme and how it works. We'll go back to that picture real quick at the other slide. So in the bottom right, let's see how this enzyme works one more time. I have isocitrate, a general base, deprotonates the hydroxy group. That carbonyl collapses, and it breaks the bond between the carbons 2 and 3, shown as 1 and 2 in this picture. Right? It grabs a proton from some general acid. We'll see what those amino acids are, or those general acid and base donors are in a second. And that releases our glyoxylate and succinate as our products. Okay. Same reaction shown here in reverse, since you can run it either direction in a test tube. We have glyoxylate and succinate, and it's much easier to run it that way in a test tube. So if we have a general base this time, which is our acid in our normal mechanism, it deprotonates the succinate, collapses to a carbanion. Carbanion attacks the carbonyl, of course, and forms the O-, which then gets protonated, right, to form isocitrate. Right? So how does this reaction help or hurt anyone? Well, we don't really use it in humans. We don't have this enzyme, but some microbacterium do. Right? So this Mycobacterium TB, which is the organism that causes tuberculosis, is totally dependent on this pathway. This invader, right, this infectious organism, when it's inside the body, may be living in an area or dividing in an area where it's causing the infection that has no sugars. It's very low in sugar. It exhausts all the supply of sugar. So it needs to survive on fats, right? So if it gets into a fat area or fatty tissue, it can still survive without any sugars, but on the fats of the cells that it's invading. And so if you have a lot of fat stores, this organism survives there. Well, one way to treat that would be to inhibit this enzyme. We don't have this enzyme, so it doesn't affect us unless it has some side effect with another enzyme. But we can treat it specifically by inhibiting TB's organisms, this particular enzyme, this isocitrate lyase. Well, how does it do that? What drug did we come up with? Well, there's some drugs at the very bottom. One on the right is called 2-vinyl glyoxylate. That's what they named it. So it's a glyoxylate with a vinyl group attached. And so how is that different? Well, the mechanism is shown there for the normal reaction. Looking on the bottom left again, you have 3 molecules with enzymes A and B, or pathways marked A and B. No, sorry, not enzymes. But in the first step, again, we have a general base there that deprotonates the hydroxy group, right? Collapses to a carbonyl that releases the bond between numbers 2 and 3, right, to form a double bond between the carbons, and it goes up and forms an O-. That's a very unstable, right, doubly charged enol form. So in the enol form, of course, collapses back to a carbonyl, forming the alkene, right? The carbonyl, which will then grab a proton from a general acid. In this case, it's cysteine 191, acts as the general acid, and it protonates to make succinate. No issue there. It's a reverse aldol reaction. When the vinyl group is present on the vinyl glyoxylate or on the vinyl isocitrate, which would be in the same place in the other end of the reaction, we have a different thing that takes place, right? So that's shown on the final slide, which comes from this paper at the top. Our normal reaction takes place, I'm just going to point to things in boxes, right? You don't have to worry about all the others right now. Just look at the things in boxes. In the normal reaction, we have our substrate bind. Again, we deprotonate, shown in that top box. We deprotonate the hydroxy group. It collapses to a carbonyl. We break the carbon bond, and it forms an O- on the top. No big deal. That then collapses, right, to a carbonyl, grabs the proton off of the cysteine, and we have succinate that's released. Nothing different than normal. On the other side, when we release the glyoxylate, it is now a vinyl glyoxylate, or we just add vinyl glyoxylate to start with, if that's the direction we're describing it. And it's coordinated by the magnesium ion there. However, because it has that vinyl group, and the vinyl group is quite reactive, instead of the succinate deprotonating the cysteine, the vinyl group forms a covalent adduct to the cysteine. So we've effectively added a molecule covalently to the cysteine, and it's never coming off. So this acts as a covalent bond, which is an irreversible inhibitor. So without the vinyl group, we won't have this problem. But this occurs occasionally. It's not every time. It's about 15-20% of the time it'll form this adduct, according to the paper, and it inhibits the enzyme. Of course, if it doesn't form the adduct, and we end up with this product, it can always go back and bind in the active site again, and eventually form the adduct. So this is an irreversible reaction. So it'll inhibit the enzyme, and of course, we'll have to make a new one. But that means this drug is also used up. It's a suicide type inhibitor. Okay, so to summarize what this is doing, if you look at the structure of 2-vinyl glyoxylate, it lets the reaction take place as normal, right? Most inhibitors don't do that. This one lets it take place as normal, except it now binds to one of the donating or either if it's acting as a general acid, it takes the proton away, or if it's acting as a general base, we'll form an adduct to it so it can't act as the acid. Right? If you look at 3-bromopyruvate on the left, it is very similar in structure to the 2-vinyl glyoxylate. Just has a bromine there instead of the vinyl group. So the bromine could get attached to the sulfur instead, right? And that's not going to come off. All right, so you could attach all sorts of things there. You probably could use a mercury compound, right? Because the mercury would definitely attach to the sulfur. However, they probably didn't pass clinical trials with a mercury type compound because it'll probably bind to lots of sulfurs on lots of enzymes. So it's not so selective as this one is.